All right, everybody. I know I am not the Amoeba Sisters. I am not that tech savvy. But what we're going to look at today are viruses. So you have a note packet. It should say viruses going down the middle of it. So we're going to go through this real fast. What you're looking at here in this picture is actually the HIV virus. And when we talk about viruses, all cells have proteins on the outside of them. And I always use shapes to represent those proteins. But these little things you see sticking up are proteins coming off of that virus. And it actually uses those proteins to stick to your cell. And what viruses do is every virus has a different protein on it. So you can think of every virus as having a shape. So you can think of this one as having like little circles, or like another virus might have squares on it, another virus might have triangles on it. And they're looking to find the cell that matches. So if you have if this guy has a protein that looks like this, it's looking for a cell that has a protein that looks like that, like a C shape, so it can stick to it like a piece of a puzzle. And so different viruses will stick to different cells. And so that's what you're seeing here in this picture. Okay? But in the case of viruses, we've been talking about the kingdoms. And you guys know that there's 6 kingdoms. 2 of them are bacteria, archaebacteria and eubacteria. And then you have plants, animals, fungus, and protist. Well, viruses aren't in any of the kingdoms. So if you're asked, what kingdom do viruses belong to? There's no answer. Okay? Because to be classified, you have to be considered living. I remember, to be living, you have to have a cell membrane made of lipids. You have to have cytoplasm. And viruses have none of these things. So viruses are considered to be non-living. Right? And so here's another picture you can see where I use different shapes to represent the proteins on the outside of them. Sorry that it's jostling around. I'm using an iPad to record this so I can put it on YouTube so you guys can see it. Like I said, Amoeba Sisters, man. But viruses are non-living particles. They're not made of cells. Remember, to be living, you have to be made of a cell. And cells have cell membranes, they have cytoplasm, they have ribosomes, and they have DNA. Well, viruses cannot be called cells. They do not have a cell membrane. They do not have cytoplasm. I always consider viruses to be like M&Ms. Okay? Some viruses are chocolate M&Ms, and some viruses are peanut M&Ms. But you guys know that M&Ms have a shell on the outside of them. Well, all viruses have a shell on the outside. And depending on what proteins make up that shell, that's what determines what kind of virus you have. So, and some viruses have DNA in them, so they would be like your chocolate M&Ms. And some viruses have peanuts in them, so that would be like your RNA viruses. But all viruses have, or you might, when you guys were little, you might have put money like in a slot and you got out like one of those bubbles and had like a toy ring in it or something. You know, like that bubble of plastic that you had to crack open, and it had one thing on the inside. Well, that's what viruses are like. Okay? They are just like this protein shell with either a string of DNA or a string of RNA. There's nothing else in them. No cytoplasm, no goo, nothing. So that's why we can't call them living. And since they don't have anything in them, they can't carry out metabolism. So they cannot feed themselves. They can't replicate themselves. They have to have help. So they are able to replicate. We know they do because they're still around. You know, they haven't died off. But they can't do it by themselves. They can't do mitosis by themselves. They can't do meiosis by themselves. Again, they're just an M&M. So what they have to do is they have to be parasites. What the virus will do is the virus is going to use these shapes to hook onto a cell, and then they take over that cell and they tell that cell to make baby viruses. And so they have to have what's called a host cell. Now, all DNA, like I mentioned, have a shell on the outside of it. Okay? All DNA. All viruses have a shell on the outside, and that shell is this thing called a capsid. What you're looking at here in this picture is a classic virus. It's called a bacteriophage, and we actually use this virus in a lot of experiments because it infects bacteria. It doesn't mess with humans, so it's a real easy bacteria to do experiments with. But I keep saying bacteria, I'm sorry, virus to do experiments with. But all viruses have this shell on them, so that would be like the shell of the M&M. And that shell is called a capsid, and the shell is made from proteins. And so different viruses have different kinds of shells. It's like the flu virus has one kind of protein in its shell, while mono, which is another virus, has another kind of protein in its shell. HIV has another kind of protein in its shell. So different kinds of viruses, their shells are made up of different proteins. That's what makes them different. And then they'll either have one piece of DNA or one piece of RNA inside of them. So when we talk about viruses, they're not classified, but they're just put into two groups. You're either what we call a DNA virus, which means you have DNA in you, or what we call an RNA virus, which means you have RNA in you. But like I said, it's just like having a piece of plastic with a string inside of it. You either have a red string or a blue string in you. So if you have a red string, you've got DNA in you. If you have a blue string, you have RNA in you. And there's nothing else in there. Okay? So another reason why we cannot call them living. And so this is what I was talking about with the shapes. Okay? So depending on what kind of proteins make up the shell, that determines the shape of the virus. And they have to fit together. They have to attach to the host. So you can see all these different shapes that they have. Okay? And so think of that as Velcro. So they have to be able to stick to their host. And so let's just say, let me just bring this in. So you can see how this guy has little circles. Okay? So it's looking for a cell, or it's got circles and triangles in it. It's looking for a cell that it can stick to. This guy has a different type of shape. It's going to stick to a different kind of cell. This guy down here has a different shape. It's going to stick to a different kind of cell. But what viruses have to do is they have to be able to stick to our cells. And they use their proteins like Velcro to help them stick. Okay? But like if I give you the flu virus and I put the flu virus in your stomach, nothing's going to happen. It can't stick to a stomach cell. But if I put the flu virus in your lungs, that's the cell it's looking for. It's going to stick to your lungs. It's going to infect your lungs. And when we talk about an infection, what happens with a virus is the virus wants to make baby viruses, but it can't do it by itself. So it's going to get in, it's going to stick to your cell, and with a needle, it's going to put its DNA or its RNA into your cell. And your cell's going to go, excuse me, hey, what's going on? And the virus says, hey, you know what? I need some help. I need to reproduce, but I'm kind of broken right now. Can I borrow your ribosomes for a minute? My ribosomes aren't working. It doesn't have any. So can I kind of borrow yours for a minute and make some proteins? And your cell's stupid enough to say, yeah, sure. But when your cell starts making proteins, it's making baby viruses. And then your cells will make these baby viruses very much like a factory. And then the more baby viruses you make, the cell starts to swell, and eventually the cell explodes. When the cell explodes, all the baby viruses come out, and they start this all over again. So depending on the shape of the protein, depending on the shell it has, that's going to determine the type of host. And the host is whatever cell it's going to stick to. So like I said, meningitis sticks to brain cells. Hepatitis sticks to liver cells. Influenza, which is the flu, sticks to lung cells. So different viruses will only stick to certain cells, and it's all based on the proteins that they have. And so like I said, that's what this talks about here with these things called host cells. Viruses are species-specific and cell-type specific. So there are some viruses out there. For example, you guys have probably heard the swine flu. The swine flu used to only infect pigs because pigs have some proteins that we don't have. So if I gave you the swine flu virus, you wouldn't get sick because we had no cells that it could stick to. Or what's called the tobacco mosaic virus. The tobacco mosaic virus gets its name because it only infects tobacco plants. Well, tobacco plants have different proteins than a human. So if I put that virus inside of you, that virus can't find any cells to stick to, so it can't do anything. So that's what we mean when we say species specific, because different creatures are going to have different proteins. So a virus that infects humans can't infect a plant because our proteins are different, our cells are different. So if you put the flu virus in a plant, it can't find anything to stick to, so it can't cause an infection. Now, HIV is a whole different ballgame. Okay, think about DNA. Usually we take DNA, and then we take the DNA and we make a photocopy called RNA. And then we use the photocopy to make the protein. Well, you have some things out there called retroviruses. Okay. The root word retro means backwards. These viruses work backwards. They do not have DNA in them. They have RNA in them. So what they have to do is use our cells to make DNA. Remember, our cells take DNA and make RNA. Well, retroviruses use our cells to take their RNA and work backwards to make DNA. And that's why things like AIDS is so bad, because our brain is like, our body is like totally confused. Because our body's like, whoa, what are you doing? You're supposed to take DNA and make RNA with it. Why are you working backwards? So these things are what we call retroviruses. They are what we call RNA viruses. They just have one piece of RNA in them. And what HIV does, this is HIV here, so you can see the different shapes it has. It will use those shapes to stick to your cell, and then they will inject the RNA into our cells. And they'll use an enzyme called reverse transcriptase. Remember, anytime you see ASE at the end of the word, you have an enzyme. And the name of the enzyme always tells you what it does, and you read it backwards. So this enzyme does transcription in reverse. Remember, transcription is where you take DNA and make RNA from it. So reverse transcriptase is going to work backwards. It's going to take the RNA and use it to make DNA. Right? And so HIV infects your white blood cells. So let me just take a second to talk about your immune system a little bit. Every person in this room is different. So every person in this room has different proteins. And your immune system knows what proteins to look for. They're what we call your marker proteins. So every cell in your body has what's called a marker protein. My marker proteins are different from your marker proteins. So what your white blood cells do, which are your immune system, they're simply going around your body looking for cells that don't have your proteins. And if they find something that's missing your special protein, they kill it. That's why people who have organ transplants have to worry about rejection. Well, your body goes, great, I got a new kidney, but yeah, it's not my kidney. So their body tries to destroy that kidney because it doesn't have the right proteins on it. Well, you have cells in your body. So think about it when you need help. You pick up the phone, you call 911. And when you call 911, the dispatcher answers the phone. And the dispatcher gets your information, and the dispatcher sends the fire department, the police department to your house for help. Well, you have cells in your body called helper T cells. They're your dispatchers. So anytime you're sick, these helper T cells say, hey, I've got an infection over here. You guys, you white blood cells, you immune system, need to come over here and kill off this infection. Well, the problem with HIV is that HIV kills these helper T cells. They kill the dispatchers. So it'd be like you needing help. There's a fire, or there's somebody robbing your house, and you call 911, but nobody ever answers the phone. Well, if nobody ever answers the phone, the police department doesn't know that there's a bad guy. The fire department doesn't know you need help. And that's what happens. People don't die from AIDS. People die from other infections that they get. Most people who die from AIDS really die from pneumonia. Because without what HIV does is it kills your immune system. So it's like you dialing 911 and nobody answering the phone. Your body never knows it's sick. So something like pneumonia, which most of us can kill off before we ever get sick, ends up multiplying and multiplying, and that's what kills that person. Now, in the case of replication, there's two ways that viruses can get inside of our cells and kill us. So notice there's two types of reproductive cycles. They both have the same first two steps. So on your page, you'll see something called the Lytic Cycle, and you'll see something called the Lysogenic Cycle. Okay. But in both cases, they have to attach, and in both cases, they have to enter. And that's what you can see happening right here in this little diagram, if it decides to move again. Okay. So you can see that there's shapes on the outside of the virus. I'm going to bring this in. And you can see that there's shapes on the outside of the cell. Those shapes are proteins. They're called antigens. Okay? So you can see what are called docking proteins. So the virus's shapes have to fit the shape of the cell. So if this didn't fit, it would just bounce right back off. But notice how it fits. So it can attach to the cell, and it can enter the cell. So now I have the virus inside of my cell. And this gives you an idea of how big a virus is in relation to your cells. So you can see again how it's got these shapes, and the shape has to match up with the shapes on the cell. So you can see how they lock in place, and then your cell brings the virus into it. So that's what we mean by attachment and entry. So on your sheet with Lytic Cycle and Lysogenic Cycle, the first two steps are the same. Now what happens after the virus enters is what makes these two types of replication different. Okay. The first one's called the Lytic Cycle. Okay. The first thing that has to happen is the virus has to attach. And that's what you're seeing right here. Okay. This is what's called a bacteriophage. This is a bacteria. Notice there's no nucleus, there's one piece of DNA, it's in a circle, it's called a plasmid. And then over here, letter B, is the virus entering the cell. It's got this little piece of DNA that's going down inside of it. So that's attachment and entry. Okay. What's happening down here in letter C is what's called replication. What viruses in the Lytic cycle do, Lytic means to pop. They will put their DNA or their RNA into your cell, and they'll trick your cell into letting it use the ribosomes. Remember, ribosomes make proteins. So your cell starts making virus proteins, and the virus kind of hijacks your cell. So your cell can't make its own proteins anymore. Your cell's making virus proteins. And what the virus proteins are pieces of a virus. So it's like a car plant. Over here's the bumper, here's the headlights, here's the engine. So you're making pieces of a car. Then you'll do what's called assembly. You'll put the pieces together. So you're going to make baby viruses. And you keep making baby viruses, and you keep making baby viruses till your cell is so big that it lyses. That means it pops. And when it pops, all these baby viruses come out, and then they'll go land, and that's how the infection spreads through your body. So with the Lytic cycle, as soon as the virus gets in you, you start getting sick. And by the time you realize you're sick, that virus has been in you for about 2 weeks. Right? So it's been killing your cells have been exploding for about 2 weeks before your conscious part of your brain starts to feel bad to let you know something's going on. But with the Lytic cycle, as soon as the virus gets in you, it's going to start killing your cells. So you're going to feel sick immediately. Now, some viruses, like HIV or mono or even tuberculosis, they do what's called the lysogenic cycle. With the lysogenic cycle, notice they still attach to your cell and they still get into your cell. But this time, excuse me, they do what's called a pro-virus formation. What happens is they knock on the door and they say, "Hey, Mr. Cell, would you mind babysitting for a little while?" And your cell says, "Sure, I'll babysit." So the virus puts its genetic material into the cell, but it doesn't take the cell over. So your cell's kind of babysitting the virus for a little while. And so anytime you make a new cell and you copy your DNA, you'll also copy the virus's DNA. But it's not doing anything yet. So it's like you're babysitting that virus. So you'll do what's called a pro-virus formation. That's where your cell takes the virus's DNA and adds it to its own. And then your cell will just go through cell division. And your cell's still happy, your cell's still doing its job, but the virus is in you. So are you sick? No. Are you infected? Yes. The virus is in you, but it's not doing anything yet. That's called the lysogenic cycle. So you could be positive for a virus for years and it never do anything. But for some reason, it says, "Okay, I'm bored." And it wakes up. And that virus then says, "Okay, cell, I'm hijacking you now." And it starts to make the baby viruses. And remember, that's what we call the Lytic cycle. Okay. So with time, diseases which show symptoms quickly are in what we call the Lytic cycle. That's when the virus gets in, takes over, starts killing your cells. You're going to feel sick. Diseases which take a long time to occur can be in the lysogenic cycle. Okay. That means you have the virus, it's in you, but it's not doing anything. It's like your cells are just kind of babysitting it.